The overall goal of this procedure is to pattern cells on silicon dioxide surfaces according to a predefined underlying design of Parylene. This is accomplished by first creating a photomask of the desired pattern. The second step is to oxidize and then coat a silicon wafer with Parylene. Next, photolithographic processes performed in a microelectronics cleanroom facility are used to selectively etch the Parylene coating to reveal the desired design. The final step is to activate the chips using first piranha acid and then incubate in fetal calf serum for 3 to 12 hours, after which a cell suspension can be applied for culture. Ultimately, cell patterning can be assessed either by live cell imaging using a dissecting microscope or after fixation using confocal fluorescence microscopy.
The main advantage of this technique over several existing cell patterning platforms is that there's no need to bring biological agents into the cleanroom. Also, pattern chips can be kept indefinitely until they're activated with first piranha acid and then serum.
To design the desired Parylene-C configuration, use a layout editor software package that is capable of reading, writing CIF or GDS2 files. Outsource photomask manufacture to an appropriate microelectronics facility, or make in-house if facilities exist. Then, oxidize a silicon wafer in an atmospheric horizontal furnace at 950 degrees Celsius for 40 minutes to produce a 200 nanometer silicon dioxide layer. After that, confirm the thickness with a small spot spectroscopic reflectometer. Place wafers in vacuum deposition system.